These are little auks, the most abundant seabird in the Arctic. They only grow to about 20 centimeters or 8 inches in length and weigh about 150 grams or a third of a pound as adults. They're migratory birds that forage for food by diving deep underwater and capturing zooplankton as they float back up. This reliance on the sea for food is part of why they serve such an important function in Arctic ecosystems. Little auks are ecosystem engineers. That is, they're a species that can drastically change the landscape around them. The little auk can not only deposit nutrients on land that they've captured from the sea in the form of guano, supporting lower trophic levels, they also act as prey species for other Arctic animals like Arctic foxes, glacial gulls, and even occasionally polar bears. The little auk has been described as crucial to the Arctic and their ecological indicators. That means that the little auk's population abundance, migration, behavior, overall health, and other metrics are all important to study because they affect and reflect the health of the Arctic. As is the case for many Arctic seabirds, the little auk contends with a number of obstacles like global warming, the retreat of polar ice, and exposure to toxicants in the oceans where they hunt and swim. One of these is mercury, which is present at rapidly increasing concentrations in the Atlantic Ocean. One paper, published in 2014 by Jérôme Faure of the University of La Rochelle and an international team of researchers from France, Canada, and South Africa, studied the threat of mercury, a well-known neurotoxin and endocrine disruptor, to little auks, and their findings encouraged ecotoxicologists to look further into spatial ecotoxicology. But what is that? And does this research indicate yet another threat to the Arctic? I'm Amara Spellord, and this is Spatial Ecotoxicology and the Little Auk. From 2009 to 2011, Jérôme Faure and his team studied a population of little auks in the North Atlantic to observe both the effects of mercury exposure on these important birds and the effect of spatial ecotoxicology on the Arctic. Ecotoxicology is, of course, the study of how toxicants flow through and affect ecosystems, but ecosystems aren't closed. Energy, nutrients, and pollutants flow into and out of them all the time. Spatial ecotoxicology is the investigation of how toxicants might flow into ecosystems because of global patterns like wind and storm systems. One of those global patterns is that of migratory animals, like many birds, who can travel thousands of miles between their wintering and summer sites, bringing toxicants into otherwise unaffected ecosystems. So what effect might the migration of the little auk, a key species, have on the Arctic? Faure et al. Had three hypotheses. The first was that mercury contamination of little auks would be higher during the winter, their non-breeding season, which they spent outside of the Arctic. The second was that mercury contamination would be related to their non-breeding distribution in foraging areas, like how far south they went and where they chose to hunt while they were there. And the third was that the mercury accumulated by the auks would affect their reproduction. During egg formation, mercury can be deposited into eggshells, albumin, and yolk. This can improve the health of the mother who now has less mercury in her body, but it's also very likely to have detrimental effects on the hatchling, which has been seen in a number of bird species. Little auks molt and thus have to grow new feathers twice a year, and mercury can bind to amino acids that form keratin, the primary component of feathers. This sequesters the mercury in the feather without significant harm to the bird, and it also provides a method by which researchers can determine accumulated mercury levels. Because little auks have a complete molt of all their feathers in September after their breeding season, and a partial molt of their head and throat feathers in April before they return to their breeding site, their new head feathers can be collected to determine how much mercury they are exposed to specifically at their non-breeding area, while body feathers would indicate how much mercury they've been exposed to during the winter. In order to find out where the birds had been while outside the Arctic, the scientists attached archival light-level geolocators to a total of 135 little auks breeding in Cap Ho, Greenland in July 2009 and July 2010. These geolocators weigh about 1.5 grams, which is only 1% of a little auk's adult body weight, and collect data on light levels that can be used to determine global position, like a sundial. The trade-off is that the data can only be retrieved if the bird and the geolocator is recaptured. The researchers were able to recapture 47 birds in 2010 and 35 in 2011, after allowing the birds to wear the geolocators for a full year. They retrieved feathers from their heads and bodies, as well as blood samples. During their recapture efforts, they also checked for any eggs that the tracked birds had laid and visited every 2 days until hatching to measure the egg size and time their incubation periods. In March 2011, they also collected birds in Placentia Bay, Newfoundland, one of the known overwintering sites for Greenland's little auks, and took blood samples from them too, so they could compare blood mercury concentrations between the winter and summer. They also used carbon-13 isotope analysis to determine where those birds had been hunting during the winter. Previous studies have shown that higher delta carbon-13 values are correlated with inshore diets for a variety of animals, including birds and mammals. So what did they find? Well, let's look at the results from the paper. Figure 1 shows mercury concentrations in micrograms per gram of dry weight, measured in both blood and feather samples. On the left, the lighter boxes show blood mercury concentrations in the summer, about 1 microgram per gram of dry weight, and in the winter, ranging from 2.5 to 3.5 micrograms per gram of dry weight. The right side, with the darker boxes, shows feather mercury concentrations in the summer, ranging from 1 to 2 micrograms per gram dry weight, and in the winter, ranging from 2.5 to 3.5 micrograms per gram of dry weight. Both of these differences are significant, confirming their first hypothesis that contamination would be higher in the winter. Figure 2 shows the negative linear relationship between blood mercury concentration and delta carbon-13 values. Note that the scale for delta carbon-13 values is negative. These were significantly correlated and show that offshore diets were associated with higher mercury concentrations. The most offshore diet correlated with a blood mercury concentration was nearly twice that of the most inshore diet. Figure 3 shows the median position of birds during the winter. Each of these points represents one of the studied birds, and the color of each point indicates the mercury concentration found in their head feathers, with green being low concentrations and red being high concentrations. There was an interesting and significant pattern here. Birds who wintered in the Grand Banks of Newfoundland and the Flemish Cap, right in the middle of the map, were much more likely to have higher mercury concentrations. The results depicted in figures 2 and 3 confirm the second hypothesis of the paper, that wintering area and foraging habitat are significantly related to mercury exposure. Figure 4 shows the relationship between mercury concentrations found in female auks' head feathers and their egg size, if they laid an egg. High mercury concentrations were associated with smaller egg volumes. But interestingly, there wasn't a correlation between mercury concentration and maternal body weight, or mercury concentrations and hatching times. The research also uncovered an interesting relationship through isotope analysis that was difficult to quantify. A change in diet after breeding. Instead of eating zooplankton, their previously known diet, it was found that little auks were feeding on krill and fish larvae, and stomach content analysis showed that they'd been eating krill through the late winter too. This diet change could be one of the reasons why there was an increase in mercury exposure over the winter. So what does this all mean? Well, first, let's summarize. Little auks were more contaminated over the winter in their non-breeding site than in the summer at their breeding site. Those that wintered closer to the Grand Banks of Newfoundland and the Flemish Cap were more contaminated than others. Foraging closer to shore was correlated with lower mercury levels. Female little auks with higher mercury contamination laid smaller eggs. And little auks changed their diet for some period during the winter. No previous study has captured this seasonal change in mercury concentrations and related it to wintering habitats, and 4 et al. Have shown that there can be drastic changes in exposure over the course of an annual cycle. It now seems clear that little auks in the Arctic are facing many threats that need to be studied through the lens of spatial ecotoxicology, especially whether or not Arctic ecosystems can be threatened by migratory birds wintering in other areas that have been contaminated by pollutants. There are numerous Arctic seabird species, all of whom could be experiencing this annual cycle of exposure. This paper also indicates the need for more research on the Grand Banks of Newfoundland specifically. Why did little auks get so contaminated with mercury there? Another important avenue for research is the effects of mercury on little auk reproduction. Little auks exposed to more mercury are investing less energy into egg production, but how this effect occurs and whether or not this effect is due solely to mercury or to exposure to other toxicants is unknown. What exactly is the danger faced by little auks? What does seem to be true is that these researchers have uncovered a vast new field of research for the Arctic, and the little auk has played a huge role in that discovery. Spatial Ecotoxicology and the Little Ox by Amara Spellworth. This paper was published in 2014, which means that follow-up studies have been conducted. If you're interested in learning more, I've included the names of a few studies here on this slide.